come anytime. All right, a couple things. You guys know we were having a review today at 1 o'clock up in the lab. How many of you are planning on going up? Okay, Matt will be taking it. And your test is due Sunday by 4.
Finished by 4.
Finished by 4. In case I decide to drive up with all the possible snow coming in, I just might do that.
Is it supposed to snow again?
Valentine's Day with
I
us.
don't know. Huh?
You're spending your Valentine's Day.
Oh, we'll go out. My husband and I are going out Saturday night.
It's gonna snow.
Where are you going?
Oh, well, we're going to start. I'm going to go to the restaurant. I'm going to bed. And
Sounds awesome.
You know that was on video, right?
oh gosh, delete that stuff. I'm glad I didn't say anything worse than I did say sleep. All right, so what we're doing is we're starting our new unit. This unit information today is not on the test for exam 1. It's on exam 2. Some of you have already written your essay, so nice job. I got 13 of them, 12 or 13 of them graded. So what we're doing today, we're starting the urinary system, which this unit, I love this unit. It's when I was in my class in exercise physiology, I had a renal physiologist, and so he was obviously really into this, and now I love it too. It's a lot of chemistry as we go along, fluid and electrolyte balance, acid-base balance. So if you like chemistry, it's a great unit. So today all we're doing though really is anatomy. And so I just want to introduce you to the urinary system, in which we know we have 2 kidneys, 2 ureters, urinary bladder, and urethra, but really the workhorse of this system is the kidneys. And we'll be talking mostly about what they do. Now, in your notes, a couple things to observe is, and we'll come back to this in just a minute, but we want to first look at what does the kidneys actually do. And so make a star by the general functions. You really need to know general functions. What does the kidney do in our body? Because many people know that it filters the blood and forms urine, but it does more than just that. And so it's a waste treatment plant to get the bad stuff out of the blood. But it also, remember from last semester, we talked about how it produces an enzyme that converts that last step forming calcitriol, which is a hormone, remember, made from vitamin D. Remember vitamin D, and then the liver makes a calcidiol, and the kidney releases the enzyme to produce calcitriol. That enzyme released from the kidney, remember, is controlled by what other hormone? Parathyroid hormone. Yeah. So the parathyroid hormone is regulating that. What does calcitriol do?
It helps with the absorption of calcium.
Nice job. It absorbs calcium, increases the absorption of calcium from the small intestine. So here we see a kidney not helping to monitor and maintain calcium levels in the blood. So the next thing it does is it causes the production and release of erythropoietin, an idea we talked about last week, I think, and that's controlling the red blood cell production and the amount of red blood cells in the blood. And then formation of ion levels and acid-base levels. What we're going to see is we can eat whatever we want, right? I mean, in terms of food, and we have certain amounts of, maybe we take in excess sodium or potassium or fluoride or calcium. It's the kidney's job to get rid of the excess. And so maintaining normal levels in blood is central to the kidney. So whenever your patient's in renal failure, they're going to have problems, right, in terms of regulating electrolytes. Also acid-base balance. We'll do a whole lecture on acid-base balance, plus a lab on acid-base balance. Because we're going to see that the kidney is able to eliminate acid and base, depending on the pH of the blood. So it's also regulating now electrolytes in the blood, and including acid and base levels in the blood. So regulation of blood pressure, it's controlling by getting rid of urine, we help to decrease blood volume to maintain blood pressure. But it also releases that enzyme called renin, that's your short essay, ultimately getting angiotensin II, which can raise blood pressure. So here we see this idea again that the kidney is really maintaining part of the cardiovascular system in terms of blood pressure, really maintaining blood as it should be. And so the last concept there, engaging gluconeogenesis. Gluconeogenesis is really only a function of the kidney if the body is in a starving condition, anorexia, or actually starving, or sometimes elderly people will have this. And so it's going to engage in producing glucose from a new source. Usually that function is controlled just by the liver and maintained by the liver. But the kidney can be an assistant to the liver in doing that. And when you look at all those functions, look at what the job of the kidney is, really, to maintain blood in a healthy manner. And so whenever you have patients, and you will have patients with renal problems, you gotta think about what all is the kidney doing for this person if they're no longer able to do it. Right? And so we'll talk about some key things as we move along today. All right, so here we are, 2 kidneys. A couple things we'll notice, and I mentioned to you before, is they are not completely protected by the rib cage. That's important. There's an illegal punch in boxing, some of you may know, called a rabbit punch, where you come behind the other boxer and you try to actually put him in renal failure. That's why it's illegal, because it's only partially protected. The other thing that we notice is that the right kidney is slightly lower than the left. That's something you should understand, and that's because our liver is on our right side. The largest internal organ that we have has taken up a lot of that space in the abdominal cavity. So the kidneys get pushed down a little bit. Now, you're going to need a clean piece of paper. We're going to make several drawings. I think you'll probably need, if I remember, about 3 pieces. You want to get them all out at the same time. So we want to look at, this is just a photo I should get right there, showing the minor caves, major caves, renal pelvis, the ureters, and then leading down to the bladder. I think so too. Now, if you want it, that photo, just send me email, I'll send it to you. I just didn't figure you needed all that color in your mix. All right. Here's, we're going to draw the kidneys and the way that they're protected in the abdominal pelvis. One of the things I should have pointed out to you before, I'll go back to this figure just a second. There's a place, see how the kidney's slightly indented here? This is called the hilum. Hilum is a word we're going to see again. It's sort of like the port of entry. So the ureter's coming out at that point, the renal artery, renal vein, the nerves, the lymphatic vessels, all coming out at this point we call the hilum. Now, what is the layer directly adhered to the kidney?
Visceral.
Now, is it a visceral layer? And if it's not, why isn't it? Is it a visceral layer? Why is it not a visceral layer? Because the kidneys are located where? Retroperitoneum, right? They're outside the parietal peritoneum. So a visceral always has to be inside the parietal layer, so it's not. Instead, what we see is what layer here? So you took it off when we did our dissections on the kidney this week in lab. Directly adheres to it. Joe?
Fibrous capsule.
Fibrous capsule. Fibrous capsule. Now, the fibrous capsule is sometimes called the renal capsule. It's called that because it's made of fibrous tissue around the kidneys. Now, around the fibrous capsule, we're going to see a layer of fat, adipose connective tissue. And since we've heard this prefix a couple times now, peri, P-E-R-I, means to wrap around something, like we saw it with the pericardium, we saw it with the peritubular capillaries, we're seeing it again. This is called the perinephric fat. And nephron, we know, I think a little bit from lab now, that neph has to do with the kidney, right? As well as the prefix renal, or the word renal, also kidney. So it means around the kidney, just fat around the kidney. Then we have some fascia. Fascia is like a really tough spider web made of areolar and dense irregular connective tissue. And this dense spider web, if you will, tough spider web, is anchoring the kidney, putting out these protein fibers in different directions to help anchor the kidney in the abdominal cavity. So this one's called renal fascia.
And you said it's dense connective?
It's dense connective tissue in areola. And then we have another layer of fat. So the kidney is quite insulated by fat, 2 separate layers. And And this one is called Cara. No, right. You just have to be careful with the spelling. That one's Cara, that one's Perry. And both levels of layers of fat are there to support and insulate the kidney. We talked, when we did tissues last semester, about adipose connective tissue, and we had structural adipose connective tissue. This is an example of structural adipose connective tissue that we really don't normally use for energy. In fact, it can cause this problem called ptosis. If a person is starved for fuel, they'll go in and take it from there, and what will happen is, this is helping to anchor the kidney to the abdominal wall, and the kidney may fall, called ptosis, the P sign, and cause the ureter to kink, and then the urine backs up in the kidney, can induce renal failure.
And that's specifically of the paranephric, or could also be the
It's both.
paranephric?
Yeah, it's both.
So just the, okay.
Yeah, all that back there. Now, just so you could, if you wanted to take your figure, what you could do is, in terms of where the parietal peritoneum actually is located, it's running like this. So that's the parietal peritoneum. So the kidneys we see are posterior. So this is the vertebral column. That's the anterior. This is the pancreas. We'll be talking about the pancreas is also in the retroperitoneal space when we get to the digestive system. Everybody good with that? There's a learning strategy too that shows you here exactly where that line would be.
So that included that in circle B, right?
It would look just like this one. Yeah. I think you guys have, yeah, this one's in your
Yeah,
book.
that's what I'm at.
All right, that's good. Got that? Renal ptosis we talked about. This is an image of a cadaver, a kidney of a cadaver. And it's kind of cool since we've already gone to lab. You can see much better than we did on the sheep kidney. You can see the cortex and the renal lobes and the minor and major calyxes and the renal pelvis and the ureter. Very nice, and even some renal pyramids up there. What we want to do is we want to draw it ourselves. So this is our second drawing. So we're going to leave that one up with the protection around the kidney. Attempt to draw in the same orientation as we have that one. Everyone's going to lab this week. So this should look very familiar. And so what's the outer portion of the kidney here called? So it's a region, right? The outer region for the renal cortex. And then the inner region here where the pyramids are. Now, in the renal medulla, we have these renal pyramids. Typically, we would see anywhere from like 8 to 15 renal pyramids. The tip of the renal pyramid, the apex of these pyramids, is what? The renal papillae. And remember, in those renal papillae are the papillary ducts. So it's kind of hard to show these areas, but if we were, this is all space for drainage of urine. So if we look at that space that's there, the cavities, from sort of draining each pyramid, what's that area? How many minor calyxes do we have? Just so you know, by the way, calyx is singular, calyxes, plural. 15. So we have the same number, whatever number we have pyramids, renal pyramids we would have those those number of minor calyxes, because they're what drain each pyramid. And then we'd see those drain, several of those would drain into a major calyx, and we have typically how many of those? 2 to 3, and then ultimately one renal pyramid, one renal pelvis. And so we can see that much better, even if I go draw it, I can, but we can see better here the minor calyx associated with each pyramid. And then when the pyramids, when these minor calyxes are draining into one place, like here, it's a major calyx. So we see there'd be some coming up from underneath and this way. And then ultimately they drain into that one renal pelvis. Ultimately then, of course, into the ureter. All right, the only thing, go ahead.
So when urine drains, it just straight goes into the ureter. It doesn't have like there's no like valve or anything stopping.
Oh, that's a good question. No, it's continuous movement. Yeah. Now, we have on the lab, and so we know these two structures. So we don't. I want you to write this for now. The bottom of the page, which is page 4 of your notes, talks about the innervation of the kidney. The kidney is innervated by the sympathetic division. It's also, by the way, innervated by the parasympathetic division. We just don't know what that does, so we just ignore it. But there's 2 places that it's going to innervate. One is the juxta glomerular apparatus. Remember, that's the thing that that's the structure regulating blood pressure. And it also innervates both the afferent and the efferent arterioles. And we'll see next week what the sympathetic nervous system does to those 2 structures. But since we've gone to lab, at least you have an idea in mind what they are. All right, we're now ready to move to drawing the nephron. Okay, so as I pointed out to you in lab, the nephrons are the microscopic functional unit in the kidney. We have millions of those, and they've attempted to show us where these nephrons are positioned relative to the cortex and the medulla. And so when we draw this, we're also going to add this to it. But here's the arcuate artery that we talked about in lab. So this sort of all the curly part, what I like to think is the convoluted part, the curly part, is all up here in the cortex, where only these straight tubules are down here in the medulla, making the pyramids appear striated. So our third drawing is going to be the most important drawing that we're going to look at. So we're going to be spending a good bit of time on this drawing. So if you take a clean piece of paper and you turn it sideways, it'll probably fit the best. So we're going to begin with the afferent arteria. And then we have the glomerulus that it leads into. That's a capillary. So an arteriole leading to a capillary is nothing new to us, right? But what's unusual is that glomerulus, that capillary, is then drained by the efferent arteriole. We would expect it to be a venule, and it's not. There's no exchange there, it's just filtering that's taking place. The afferent arteriole, I want you to note, is larger in diameter than the efferent arteriole. Can you read this, Joe?
Yeah.
Because it is cursive, a little bit of cursive there. I love that. Now, what we're going to do is we're going to take and add to this figure the glomerular capsule.
Really testing my drawing skills today.
Yeah, mine too. Alright, So this is encapsulating the glomerulus, so it's called the glomerular capsule. Glomerular capsule. So remember, glomerular capsule. And the glomerular capsule ends up actually with two layers. We'll identify them as 1 and 2. And so the inner layer is the visceral layer. That's number 1. And the visceral layer is permeable. It's going to allow things to leak through it. And we'll be talking in a lot of detail about its specific anatomical structure next week. And the second one is the parietal layer. And the parietal layer is impermeable. It's actually made of simple squamous cells, and its job is to catch the fluid. Right? In this space here, called what? Capsular space. So the capsular space is here. And the fluid that gets caught, if you would, caught by in the capsular space, that fluid is called filtrate. Why is it called filtrate? Is because it's filtered. It's filtered plasma. It's only called filtrate when it's in the capsular space. Now, these two together, both the glomerulus and the glomerular capsule, form what combined structure? On the right track. Renal corpuscle. We got the renal corpuscle. Corpuscle means body. So they thought they looked like little bodies in there, round bodies. So renal corpuscle. It's the combination of both the glomerulus and the glomerular capsule. Now, we're going to do a whole lecture on one Tuesday next week, if everything goes well, let us know, about what happens right there, how the plasma is filtered and then goes in. Now, from there, it's going to move into this next area. Just do a little bit of convolution, but this is called what? Proximal, because it's close to the glomerulus. Proximal convoluted, because it's not straight, tubule. Abbreviated, the PCT, which you won't use, all right, in your lab. You'll be telling me it's the proximal convoluted tubule. Now, the proximal convoluted tubule is a very relatively simple tube. It's made of simple cuboidal cells, not much of a barrier, one layer of cuboidal cells. And the fluid in here is now called, once it makes its way into the tubule, it's now called tubular fluid. Now those cells, we're going to see in next week's lecture on Thursday, that once the fluid is filtered and comes into the tubules, it's got to be processed, if you will. And so these cells, you know how epithelial cells can have modified surface features of cilia or microvilli. What do these have? These we're drawing in here microvilli. And so microvilli, if you recall from last semester, are to increase surface area for absorption. So we're going to see, yep, a lot of absorption is going to take place there. Now, once it goes down, what's this I'm drawing here? Nephron. Patients do the drawing. it's still a single, the tube is still simple epithelium. So what's the fluid now called as it's moving down through here?
The urine.
That's a good guess, but it's still
Tubular fluid.
tubular fluid. Because it's still tubular fluid because it's still in the tubes. Yeah, still in the tubes. Now, this loop is called collectively nephron loop. And the nephron loop has two limbs, right? So we'd see the descending limb on this side and the ascending limb. Now we're going to see when we have these long loops of Henle, that loop is going to set up a concentration gradient. We'll talk more about that later. But its whole function of if it has a long loop, just write and make a note, establishes, maintains concentration gradient. Outside the loop, right, in here. It's kind of weird. It'll make more sense hopefully later. All right.
Establishes.
Concentration gradient. So we're moving the fluid, fluid's moving along, down through the nephron loop, then we get to the other side of the loop. We're going to see we're distal away from the glomerulus, so this part is then called distal convoluted tubule. Abbreviated what?
DCT.
DCT. Still tubular fluid, right, as it's moving through here.
Is there microvilli and all of that too?
That's a great question. The most microvilli are in the proximal convoluted tube because that's where most reabsorption is going to take place. These still have some sparse amounts of microvilli, but not nearly what we see in the PCT. That's how we can tell them apart on a microscope. Because there's some, but it's real, very little.
Now, within the loop, is there microvilli? Then what you would call the loop as opposed to the PCT? I mean, the proximal.
I don't know the answer to that. I don't know. I've never seen anything that describes microvilli in the loop. So I would guess no, but I could be incorrect. I'm just basing it on the fact that I've never seen it. All right. Now, our boundary for our nephron, which is what we just drew, does not include the afferent arteria or the afferent arteria. It begins here at the glomerulus. We've drawn an imaginary line there. And it goes all the way to the end here at the distal convoluted tubule. That is a nephron. So if I said to you then, okay, what are the structures that compose a nephron? You'd say, well, it's the renal corpuscle, which is made up of the glomerulus and the glomerular capsule, and it's made of tubules. Proximal convoluted tubule, the nephron loop, and the distal convoluted tubule. So two parts of the renal corpuscle, and then three sections of the tubule make up a nephron. Now, once it's there, these nephrons, numerous nephrons, will be dumping their secretions, their tubular fluid, still tubular fluid, by the way, into what's this next region called?
Collecting tubules.
Yeah, so this would be a collecting tubule.
Analysis.
Now, what we'd see is we'd see numerous collecting tubules ultimately dumping their secretions into what?
Collecting ducts.
How you doing out there with your drawing?
I ran out of space.
Did you run out of space? Start on you. Just do it. Yes, that's okay. If you have a new piece of paper, just put it on that one, and then you can tape it to it later. That's what I end up having to do sometimes. So this is the green that we've just drawn is the collecting duct. The collecting duct, ultimately, collecting ducts will drain numerous of those will drain ultimately into what duct down here. If we went far enough down,
Papillary duct.
capillary duct. So ultimately, when, by the way, the tubular fluid, still tubular fluid as it moves through here, okay? But once it leaves the collecting duct, at that point, we would call it urine. So it's not urine until we've completely processed it through the collecting duct. So when it enters the papillary duct, that would be urine, and then obviously into the minor calyx. Because it's going to go papillary duct, remember, then minor calyx, based on our working lab. All right.
Oh, terrible.
Huh? Now I'll let, if you want to take a picture of mine when we're done, I'll be glad to let you do that. Now, what we've got to do is we've got to draw an imaginary line that separates the cortex from the medulla. What part of the nephron is in the cortex? What part of the nephron is in the medulla? So is the glomerulus and the glomerular capsule in the cortex?
Yes.
So we would have it like somewhere along like this. So here's cortex, renal cortex. Here's renal medulla. So that's what we're seeing here too, right? Here's the corpuscle and the convoluted tubules up in the cortex. And here's the nephron loop and the collecting ducts, and obviously some collecting tubules down in the medulla. That's why, remember, the pyramids look striated, because these tubes are all straight. They're all going down. Except for that little loop there. Now, we've got to add a few more, if that wasn't enough. We've got to add a few more things to our figure. I know, aren't you glad it's just anatomy today? Just wait till next week. No scaring you, all right? So you got these specialized cells in the collecting tubule and the collecting duct. There's specialized cells in here, and they're the most prevalent. So these cells are called principal cells because they're the main one. So a principal cell has, as part of its in its cell, associated cell, it has receptors for two hormones. Which two hormones? One of those, Sandra's right, is ADH, which stands for antidiuretic hormone, and also aldosterone. Now, what that means is that, am I telling you that ADH and aldosterone come from these cells?
No.
No. What they're doing is they're binding those hormones. So these really serve, remember, as targets, target organs or target cells, if you will, probably a better term. These are target cells for those two hormones. In fact, where does an antidiuretic hormone come from in the body? What'd you say? Posterior pituitary, controlled by the hypothalamus. Where does aldosterone come from?
Adrenal cortex.
Adrenal cortex. Part of your short essay you were writing. Now, we have another one. We have one additional type of cell associated also with the collecting tubules and the collecting ducts. We'll make them black. We don't have nearly as many of these as the other ones, but we've got some. So these are called intercalated cells. Now, if you're thinking, you might be like, oh yeah, intercalated. I remember intercalated disc in cardiac muscle. Remember, it holds the cells together. No relation. Right? These are intercalated cells, specialized cells in this part of the tubules, and they are of two types. We've got type A, and we've got type B. Now, all I need you to know for now, just to start thinking about and keeping all these parts straight, is type A, in general, is going to secrete acid. So A is for acid. It's telling you what the cells are gonna secrete. So we secrete in order to excrete. These guys wanna get rid of acid. So when you think type A, think A stands for acid, that's what these cells wanna get rid of. So obviously then type B secretes what? So it secretes the base. Now that, if we know just that, we know a lot, because if I say to you now, most of the time we eat more acidic foods than we do basic foods. We produce more acid than base, like keto acids, lactic acid, phosphoric acid, all this acid's being made all the time in our body. The kidney's job is usually for those type A cells to be active and dump acid, right? That's normal. Then I say to you, you got a patient with renal failure. What am I concerned about?
pH level.
pH level, or in which direction? Are they more likely to go in acidosis or alkalosis? Are they more likely to have too much acid or too much base?
Too much acid.
If their job is to get rid of acid, and their kidneys aren't functioning, acid's building up in the body. It's called metabolic, one of the causes of metabolic acidosis, and they can die from metabolic acidosis. Simply from those cells aren't functioning, when the kidney's not functioning. Right? We'll have a whole lecture in lab on all that. Now, we got one additional thing to add to our figure. It should be looking pretty complicated on the paper. So when the afferent arteriole comes over here, it's going to continue into another capillary. Now this capillary, I'm drawing it like it's straight across. It's not straight across. It actually wraps around the tubules. We're not drawing it that way, right? So we're just laying up top. So instead of actually wrapping. So what's it called if it's around the tubules? Peritubular capillary. So the peritubular capillaries are the capillaries that are positioned around these tubules. The proximal convoluted tubule and the distal convoluted tubule mainly. We have another set of capillaries that run along the nephron loop. They look like, they thought they looked straight, and they were vessels, so they called it the vasa. What's the word for straight? That's the vasa. So we actually have two sets of capillaries, and normally the blood goes into one or the other. Peritubular and vasa.
Can I ask you a question? Just on wording alone.
Just on what?
Wording.
Uh-huh.
For type A and type B, it says secrete acid and base, and in the notes it says eliminate.
Okay, so you secrete in order to excrete or eliminate.
Okay.
Okay. So ultimately, we secrete it into the tubule. It's actually we'll see that acid's coming from the blood. We're going to secrete it in here, and then whatever's ends up in the urine, it's going to be excreted or eliminated.
Okay.
That's a good question. That, if you know that anatomy, that really is going to help you next week. Okay. So we've seen it in lab. We've got our drawing. So if you go over to page 8 of your notes, let's make sure I haven't forgotten anything. So we got the afferent arteriole bringing blood into the glomerulus, and the afferent arteriole bringing out. The glomerular capsule is composed of a visceral layer, parietal layer, and then we have the capsular space between them. The renal tubule has 3 parts: the proximal convoluted tubule, the nephron loop, and the distal convoluted tubule, all which we talked about. If you go over to page 9, we did not look at the 2 types of nephrons. So it turns out we have millions of nephrons, but they classify them in 2 categories based on the relative position in the cortex. So we have one group called cortical nephrons, and cortical nephrons sit high up in the cortex, towards the edge of the kidney. So they're up high, and they tend to have a short nephron loop. Approximately 85%, depends on your resource, what you'll see, but about 85% of these would be considered cortical nephrons. In comparison, the other type of nephron, which makes up about the other 15%, is called juxtamedullary, not to be confused with juxtaglomerular. This is just telling you what it's next to. Juxtamedullary is next to the medulla. It's not in the medulla, it's right next to it. So it's down low in the cortex, next to the medulla. And these tend to have the long nephron loops that extend way down into the medulla. And those are the ones that are important in establishing the concentration gradient. So it's these cortical nephron loops that establish and maintain the concentration gradient. gonna see later is that there's literally a gradient of ions out here that becomes more and more concentrated the further you move down. Outside the loop, outside the blood vessels. So it's in interstitial space, it's between the loops and the vessels. Why I make a big deal about that is because that concentration gradient must be there in order for ABH to function. So the function of antidiuretic hormone is requiring that concentration gradient. How many of you have ever heard of loop diuretics in treating high blood pressure? Yeah. That's the loop they're talking about, right? There's nephron loops and loop diuretics. All right. I think we got all that. That's good. That principal cells and the plated cells, we talked about that. All right, so our last major drawing that we're going to do is going to be of the juxtamedullary nephron. Now, here's the challenge. When we draw a nephron, we draw it as if it moves from this side one direction all the way over and it's laid out flat. Or not, right? You got to imagine that this is a piece of paper, and you can take your paper and you flip it over. The distal convoluted tubule in actual position is next to the afferent arteriole. So that, remember, the afferent's the one bringing the blood in. The DCT is the last part of the nephron. Those two have to talk to each other, right? So as the blood comes in, and as the tubular fluid leaves the nephron, these two are side by side. So that's what this is going to be right here. This is the distal convoluted tubule part of it, and here's the afferent arteriole. That's what we got to draw. So where these two abut, where they sit next to each other, and we can see this a little bit better perhaps on this figure. Here's the afferent arteriole. Here's the proximal convoluted tubule. There's the nephron loop. There's the distal convoluted tubule. So we see in this figure that you see how it's flipped back on itself, so the DCT is right next to the afferent arteriole. Very important to know those two sit beside each other. All right, now, what we've got to do is we've got to draw and talk about this structure. So, we're not going to draw it. If you guys are okay, we're just going to label it on this figure you already have in your notes. So, we have which two structures are of interest? Remember, afferent arteriole and the DCT, which stands for the distal convoluted tubule. Now, where are they? So you imagine now that this marker is the afferent arteriole, and my finger is the distal convoluted tubule, like it's an empty open tube. These sit next to each other like this, and so we end up with cells of the afferent arteriole, like this, sitting next to the tubule here. So this region in here, where those two cells come together, sets of cells come together, is a structure. All right? So this here, 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 like this. And what is that structure called? That's the juxtaglomerular apparatus. Where does it get its name? It's next to the glomerulus. So the apparatus next to, so you make yourself a note, it's the apparatus, the structure, if you will, next to, right, adjacent to the glomerulus. That's how it got its name. Now the juxtaglomerular apparatus's job is to monitor blood pressure. That to me is just weird. All right, think of blood pressure, why wouldn't somewhere else in the body? Lungs, I don't know, next to the heart or something. But here we have the kidney is monitoring systemic blood pressure. And in response to a decrease in blood pressure or sympathetic division stimulation by the sympathetic division, what substance is released?
Renin.
And renin is a hormone or an enzyme?
Enzyme.
It's an enzyme. So we have this apparatus, and its job is to monitor blood pressure. So the question becomes is how does it do that? So, well, we already know about blood vessels. We know that there's these cells in here of the afferent arteriole. They're part of a blood vessel. So how does a blood vessel measure blood pressure? Stretch. So how would these cells know if there was a drop in blood pressure? Less stretch or more stretch?
Less stretch.
So they're monitoring for a decrease in stretch. So when they detect a decrease in stretch, simply because there's less blood pressure, less blood volume, whatever the reason, there's less stretch, those cells release renin into the bloodstream.
The granules.
Those, and I'm glad you mentioned that. Those cells specifically are called granular cells. They used to be called juxtaglomerular cells, if you ever see that, JG cells.
And they monitor for the decrease in
So they're specifically monitored decrease in stretch. So you say, well, that's great, right? Why do you need the other cells of the distal convoluted tubule? Now these cells, of course, have a name too, and they're called the macula. Remember the word macula, like in the eye, macula lutea. We're seeing this one as macula densa. Tightly, densely packed tall cells like this. And this region here, they're very specialized, and they're densely packed, macula densa. Now you think to yourself, well, what the heck? These cells are part of this DCT down here somewhere. What the heck? How can they possibly measure blood pressure? All I want you to think about for just right now, write your notes, is they will detect a decrease in sodium chloride in the tubular fluid. So they're going to monitor a decrease in sodium chloride in tubular fluid. And on Tuesday of next week, we'll talk about how that is an indication of low blood pressure. And hopefully, it'll make sense to you then. Everybody good? So hopefully, not too bad. So, all that in terms of the anatomy. Now, one of the things I had you do in lab, and I couldn't find it when I went to do the videotape, so I want to make sure we got it recorded.
Can I ask a quick
We
question?
had to trace blood flow into the kidney and then back out. Kidney?
What if there isn't a decrease in stretch for the granular cells, but there's a decrease in sodium chloride? Will they still signal the granular cells to emit or release the renin? Or do they both have to work together?
You'll see, I cannot think of a scenario where that would happen.
Okay.
Since you asked the question, what's going to happen is when this is decreasing in stretch, less fluid is filtered. And as the fluid moves in here, you have more time to reabsorb all the stuff along the pathway. And so by the time it gets here, sodium chloride is just one thing that it could be monitoring. It just had to be sodium chloride. And it said, oh, we had low sodium chloride. If we had low sodium chloride, what that means is way back here, we didn't make much filtration. There wasn't much pressure to drive it out there. So I can't think of any way that would happen with those two separated from one another. All right, blood flow. So you guys, let's get through this, okay? So we're going to come off what vessel? Main vessel to bring blood into the kidneys.
Renal arteries.
Come off what main vessel here? Aorta. So aorta comes in, here's the what? Renal artery, okay? Comes in, splits into what? Segmental arteries. Segmental arteries then will go between the lobes as interlobal arteries. They will then come up here and across this arch as the arcuate artery. The arcuate artery then will branch into the cortex as a much smaller vessel called interlobular. Now, once it's in the interlobular, we have to switch diagrams, right? So we have to go to the microscopic view. Alright. So, here's the arcuate, just to get your bearing. What number was that anyway? Remember?
5.
That was 5.
Arcuate was number 4.
4. And interlobulator was 5, right?
Yeah.
So here's the arcuate, here's the interlobulator. Then what? Afferent arterial, which should be, is that the numbers right?
Yeah.
6. Comes in to glomerulus, that's 7. Gonna go out, the efferent arterial. Now it has one or two choices, it's gonna go this or this, right, which is, not with that 9A or 9B. So it either goes into the peritubular capillaries or the vasa recta. And then what's it gonna do? It's gonna come back through what vein? Interlobular vein. So if we go back to that previous figure, here's the interlobular vein. So the interlobular vein, what number are we on?
10.
10. So that would be this one. Here's 10. Comes across the arcuate as what? It's the arcuate arteries, 11. Then it's going to come back through as what?
Interlobar.
Interlobar, because it's between the lobes, which is number 12. And then out what? Which is 13. And then it dumps into 15, which is inferior vena cava. Everybody good? Because y'all checked out. All right. I think I had one student that missed lab, so I want to make sure that person had it. Now let's do flow for making urine out the body. So, we can do it with this one. All right. So it starts in the glomerulus, goes into the capsular space of the glomerular capsule, proximal convoluted tubule, descending limbal-nephron loop, ascending limbal-nephron loop, distal convoluted tubule, collecting tubule, collecting duct, capillary duct, and then we go into the big picture, which is we're going into what structure? Minor calyx. So here's the minor calyx, major calyx, renal pelvis, ureter, urinary bladder. Once you got it, doesn't seem too bad. Let's just get in here. Now, the last thing we want to do, because we've got a couple minutes, I do want to introduce you to where we're going next week, and just sort of get your brain thinking about this. So we're going to see that often what we've got to do is produce urine. So we're filtering the blood to make urine, and in order to do this, there are 3 major processes that we have to talk about. We're going to talk about the process number 1 on Tuesday, and 2 and 3 on Thursday. So this first one is called glomerular filtration. And in glomerular filtration, we're going to see that's going to be happening at the renal corpuscle, in which plasma is actually filtered and forms filtrate. Then we have 2 associated processes. One's called tubular reabsorption. Now, tubular reabsorption, the key thing to remember here is that the substances you want to reclaim, you want to reabsorb them. So we're going to see they're going to be moved from the tubule and reclaimed, reabsorbed back into the blood. So if we were going to draw a line on our figure, what we're seeing, this is the direction of reabsorption. We're reabsorbing or reclaiming it. And the other one is opposite of the process, if you will, in terms of direction, is tubular secretion. And in tubular secretion, we're going to see we're going to move, substances are going to move from the blood into the tubule. Now here's the way to remember it. So that, in that case, this is reabsorption with an R, tubular reabsorption. The tubular secretion is moving it this way, yes. Right? So the key thing to remember, way you can remember, is you reabsorb to reclaim, and you secrete in order to excrete. So to excrete something, like we excrete urine, to excrete means to eliminate. So we secrete in order to excrete or eliminate from the body. So it's sort of like a conveyor belt. So we filter stuff non-specifically, and then we want to reclaim the good stuff, get rid of the bad stuff, and whatever's left in the tube is going out as urine. That'll be our whole discussion next week. We are going to stop 5 minutes early today, just because you have so much work to do this weekend. Get your test done. And we're done. So we have our review at 1, so I'll